I'd like to start by just thanking and congratulating the Danon Institute for their commitment to research and for inviting me to speak. And thank you again to Paul as well, who has organized this session. So I was asked to talk about one-carbon metabolism in general and how it affects aging. So most of what I'm going to talk about really comes from committee reports that have really addressed some of the fundamental questions of what affects folate requirements. So that's what I'm going to talk about. And some of the themes I think that are important, especially as we age, is variability, both variability in intake that's necessary to get a response and then variability in response, where that comes from and how that determines health. And I'm going to close by talking about time and money because those are also very important concepts. First, just to start, why do we have the food system we have today? We have the food system we have today because after World War II, there was a lot of hunger. And there was just this general consensus between the US and Europe that hunger had to be eliminated, and we created a food system to feed people to avoid hunger. And then we fortified that food with nutrients to avoid micronutrient deficiencies. So we have a food system that was designed to avoid deficiency, whether it's hidden hunger or caloric hunger. That food system now is costing us trillions and trillions of dollars in healthcare costs, and that's what's driving a lot of these reforms. But this food system is doing exactly what it was meant to do, and it's been highly successful in doing that. But then there's been this externality. So how do we deal with this issue now of food that is actually decreasing health span and lifespan? And I want to start first by looking at we really are what we eat, and we are what our ancestors ate. And the genomic data on this is very, very clear. So human evolution has really defined nutrient needs. If you look at the left, all human populations, as they emerged and thrived and grew all over the world, experienced constant famine. We have been optimized for the minimum to be able to survive famine. And that's why if you look at the genome, across species, mammals, you see very, very little differences in binding affinities for vitamins and minerals, essential nutrients. They're all the same because we've all been optimized for the minimum, that energetic minimum for proteins to be able to bind nutrients. That's why we can have things like RDAs and dietary reference intakes, all of these, because the population responds the same because we've been molded that way because of famine that has forced that. If you decide that deficiency isn't your goal of the food system anymore, or avoiding deficiency, if you decide it's health, that's a completely different concept genetically in terms of evolution, because human populations emerged all over the world. Those that thrived and those that expanded were able to adapt to their local food system and their local pathogen environment. So if you look at what are the two major drivers, environmental drivers of genetic variation in human populations, it's related to nutrition, food, and it's related to pathogens. Those drive genetic variation across populations, and you can see that genomically, where there's rapidly accelerated regions within the genomes. Those are the fastest evolving gene systems and networks that have been shaped, again, by both food and by pathogens. Nutrition and metabolism there has an asterisk because it does show a tremendous amount of accelerated evolution, but it also underpins those other systems as well. Those other systems, the rapid evolution is due to or is driven by those changes in genetic changes in nutrition and metabolically related genes. So when you look at this, we've been adapted all over the world differently to different food systems. When you change the food systems, genes become maladapted and you have disease. And so there's two different ways that food has driven what now we need to consider for dietary requirements. If you're worried about just being adequate for life for essential nutrients, you can take a population approach. If you look at health as your outcome, if that's your goal, you're going to see a lot of variation theoretically, and in fact you do. And that's why a couple of years ago, Francis Collins, when he was head of the National Institutes of Health in the United States, when an elevated nutrition into the director's office with his Precision Nutrition Initiative to answer the question, what do you eat to stay healthy, where he mentions in that diet-disease relationship, one size does not fit all. You get a lot of variability. Reagan Bailey, my colleague at Florida State University, wrote this and said, this is a great concept, but we really don't have the data. We don't have medical grade evidence to really set dietary standards based on health outcomes in most cases. So one of the first steps to trying to move from our expectations of food and nutrients from just maintaining adequacy to then maintaining health or preventing chronic disease was this report from the National Academy of Sciences, Medicine and Engineering, developing a framework. Well, how do we think about setting nutrient-based guidelines based on chronic disease endpoints, not based on, preventing deficiencies? And this is a very aspirational document. I was part of this study group. But some of the conclusions from that are, one, diseases of nutrient deficiencies are relatively simple in healthy populations. If you don't get enough intake, you're going to have a deficiency disease. If you look at a chronic disease, it's really complex because it's not only what you eat, but it's your exercise, it's your sleep, it's your stress, it's all those things that Paul mentioned. We also know there's great heterogeneity in populations with chronic diseases, both the penetrance and the expressivity of the disease. Few chronic diseases are affected by a single nutrient or a single pathway, so you have to consider networks, and that's really critical, and network failures, how nutrients interact in given networks, pathways, and how that connects to disease. You have to consider healthy and unhealthy individuals, because as Paul mentioned, disease starts from conception. So there's no such thing as a healthy person, there is a trajectory, and the question is, how do you soften that trajectory? When, again, you're not considering deficiency, you have to think about stress responses and immune responses. And you have to think about all of the other food components that aren't essential for life, but might be essential for health, the so-called bioactives. So it's very complex. This is the goal we all have now, is to have food for health. But it's very complex, and as you know, we have very little data. And of course, one-carbon metabolism has been used repeatedly as an example of both the opportunity and the challenge of having health as the endpoint. So if you look here, if you look at one-carbon metabolism in general, which I'll get to, it is highly correlated to a number of diseases, from anemias to birth defects to neuropathies to cancers to birth outcomes. There are a number of diseases, and some of them are linked. Those who have family history of spina bifida have some of the lowest rates of colon cancer known. So whatever's about those individuals that makes them susceptible to birth defects, protective against cancer. You also see high variability in genes and one-carbon metabolism, and those are some of the drivers that have been shown to drive that variation. So one-carbon metabolism, you have that disease, the opportunity to lower some of these diseases, but you have the issue of variability. So I'll just run through one-carbon metabolism. This is what I spend my life doing, is studying this network and all of these different branch points here. But it's pretty simple, where you have mitochondria take serine or glycine, or betaine, as was mentioned, choline, to generate formate. And you need folate for this. Formate comes out, it's condensed back on the folate, tetrahydrofolate, to give you an activated one carbon here that can go to make purines. Otherwise, you can pump a couple electrons into that formyl group to get formaldehyde here, which you can use to make thymidylate, or you can put another 2 electrons in there using both vitamin B12 and riboflavin to then remethylate homocysteine to methionine. Methionine can then be used to generate S-adenosylmethionine to serve hundreds of methylation reactions, including DNA methylation and the epigenetic effects we saw. What's interesting here is this happens in the cytoplasm, this happens in the nucleus, this happens in mitochondria. This pathway for thymidylate biosynthesis happens in the nucleus, and I'll come back to that later. So this network is highly connected, but it is also spatially, there are spatial barriers that can disconnect this network as well. And I'll touch on that. But what's important to know is that this is a true network. Folate is always limiting. Cells do not accumulate as much folate as there is folate binding protein. So they're always competing for this limited pool of cofactors. And so if you affect any pathway here through a polymorphism or through a nutritional deficiency, it ripples through the entire network. So you can think of one-carbon metabolism here as a network. There are key nodes here. There are all the folate enzymes, and there are the cofactors that are there in low concentrations bound to these proteins to make, and enzymes to make them work. You have input serine, glycine, choline. Go in. What comes out is DNA synthesis, cellular methylation, genome methylation, among other methylation reactions. If these events then affect key outcomes of proliferation rates, which can affect birth defects, genome instability, which can affect cancer, gene expression to a large degree, those epigenetic changes. So this network is highly correlated with fundamental processes in the cell, fundamental processes related to the genome. That's why it's correlated to so many diseases. Okay, so let's talk about some of the challenges with one-carbon metabolism and using that health outcome relative to a nutrient supplementation or fortification. And this is one of the greatest success stories in all of nutrition. It never really works this way, as we all know. But early in the 1960s, there were clinical observations that if a woman presented with a birth that had a neural tube defect, a birth defect, they had biomarkers of impaired folate metabolism. But there were many moms who had biomarkers of impaired folate metabolism who had perfectly normal births. But this led to then clinical trials with folic acid supplements, which had a major effect on reducing neural tube defect frequency, leading to recommendations, which nobody follows because most pregnancies are unplanned, and you have to have the exposure within 4 weeks, often before a woman knows she's pregnant. And in the US and other countries led to US fortification of the food supply, which then through evaluation was highly successful in virtually eliminating this rare birth defect. So it worked all the way through, and there are many countries now that fortify the food supply with folic acid for that purpose, for this health outcome, to reduce the birth defect. But you see that there are also countries that don't, including a lot of Europe. People have raised safety concerns because we are putting more folate in the food supply than was needed. There wasn't deficiency in the US. There was not folic acid deficiency or folate deficiency. This was about addressing a birth defect. And the concern comes down to really uncertainty. There's a lot of epidemiological associations that go both ways. So there's a lot of literature on folic acid safety, but there's no clinical grade evidence that it's unsafe at that level of fortification. Yet people worry about unintended consequences because you're targeting such a small group. So why don't all countries fortify? Well, number one is food as medicine. We're using food for a health outcome, not to maintain nutritional adequacy. And back in the 1990s, that was very controversial. It wouldn't be today. Lack of precision, you're exposing everyone, but it targets a really small population. It's women of childbearing age, women of childbearing age who are genetically susceptible to having a child with a birth defect. And that's an important distinction. If you make any mammal folate deficient, you will not get a birth defect. You will not get spina bifida. There are certain women who we can't identify who have a genetic predisposition to have the birth defect, and they need more folate than they can get out of natural food to overcome that genetic predisposition. We don't know the mechanism, which can be problematic because we really then can't anticipate unintended consequences. And it's the effective dose. A woman who is at risk for a child with a neural tube defect can't get enough folate from a natural food diet. This will also be the case in aging, as we'll see. So this really is exacerbated concerns of unintended consequences because a clinical trial is a black box. Folate in, prevention out, but we still don't know how it works. And so we don't know whether it puts other people at risk, or at least be able to make hypotheses. So what we know about NTDs is that you need 2 hits. You need low folate status, and you need to be genetically sensitized to having that birth defect. And you need both. And if you have both of these, then you can prevent the neural tube defect. But the 2 risk factors are both folate status and folate utilization. We know some of the genetic alleles, There's a polymorphism in the MTHFR gene, which is right there, which is really a gatekeeper between, does that folate go to make DNA or does it go to methylation reactions? That variant is associated with low folate status. And the reason is that it has much lower activity and actually causes a logjam here in accumulating unstable forms of folate. And so these people have lower concentrations of red blood cell folate and need more folate to maintain folate stores. But if you knock that gene out in a mouse, you will not get a neural tube defect. It affects folate status. On the other hand, there's another mouse that we created here, and there's human variants in serine hydroxymethyltransferase 1. This gene is involved in thymidylate synthesis for DNA synthesis. And there's a polymorphism here that affects the ability of this protein to be ubiquitinated and sumoylated so it can go into the nucleus. And humans who have this polymorphism can't sumoylate very well. And that's that variant protein. It doesn't get sumoylated, so not very much gets in the mitochondria. That's a risk factor for neural tube defects, not folate status. So we see here that there are polymorphisms that can affect folate status, but not neural tube defects. There are polymorphisms and environmental exposures that can cause neural tube defects, but they're dependent on lower folate status. But you need both hits, and there's a genetic and a nutritional component on both sides of that aisle. So that tells you some of the issues you have with using folic acid for a health outcome. It's dose, more than you can get out of food, and it's highly variable among individuals. Well, what does this tell us about later stages of the life cycle? And this is very crude, but you can think again of aging, biological age, or chronic disease risk there you see, on this axis versus network decay. As we age, our biological networks decay. And Paul mentioned it's probably a step function, or you can see decay rates here. Important things to recognize is that when you're born, your networks function at different levels, right? And some of that is the epigenetics or fetal programming. And then you have different rates of decline. So how can we think of this in terms of nutrition and nutrition interventions to soften that decay curve? And one of them is nutrition. And we talked again about the developmental origins of disease, where early nutritional status plays a key role in programming some of these networks and giving them different starting points here, if you will. And again, there were many examples, and Andrew Prentice here has done beautiful work in human populations in the Gambia, showing the effects of nutrition during pregnancy on outcomes in individuals and in genomic effects. So we know that nutrition interventions, especially those around one-carbon metabolism, can really program the genome for later health. But we also know, as we age, that these networks, this decline, manifest differently. Sometimes they become more, or sometimes they become less noisy. The immune system becomes more noisy. Methylation of DNA becomes more noisy. Oxidative phosphorylation becomes less noisy. You also have higher or lower output. Homocysteine, which is involved in one-carbon metabolism, levels tend to increase with age, and in fact are a signature of healthy aging. There are also context-specific. Organs can age at different rates. You can have, one of the most important aspects of aging is your stem cell populations. The secret to life is maintaining robust, high-quality numbers of stem cells in your organs because those are your renewal potential. But we know that stem cells have very different nutrient requirements. Muscle stem cells are totally dependent on serine. And as you age, if they don't get additional serine, they're not going to proliferate. And there's some wonderful work been done in that area. But we also know that there's modifiers here of this curve, the shape of this curve, both disease, pharmaceuticals, and environmental toxins. But essentially the question, I'll show examples of these, but the question we want to ask is how can we use nutrition throughout the life cycle here to really soften this aging curve and promote health? And just want to introduce the concept of special nutritional requirements. I was on this National Academy panel a few years ago, really looking at states of medical conditions where you cannot meet your nutrient needs with a normal diet. Okay, and so that's been termed special nutritional requirements. These were the examples that they showed, But aging can also be an example of that. As you age, you can develop nutritional requirements that you just can't simply meet through a food-based diet. So a couple examples of these. First, on the concept, this was really first proposed by Victor Herbert, who was at Columbia University, an old hematologist. But this idea that if you have a disease, you have disease-related etiology that then affects how nutrients function and are processed, which then can impact human nutrition. It can give you a whole body deficiency, a tissue-specific deficiency, conditionally essential nutrients, nutrients that weren't required prior to the disease, toxicities, and of course, you have to feed your stem cells, so often you have proliferation to an advanced renew in these cases. But the concept here is that you have a chronic disease that changes your nutritional requirements. If you don't meet those, you're going to get comorbidities associated with that disease simply because you're not meeting the new nutritional requirements. And I'll show you some examples of those. One is diabetes and folic acid. So diabetic neuropathy diabetes is the major cause of neuropathy in the US and globally. And of course, it is loss of sensation in the periphery. Well, there have been animal studies that have come out the last couple years, and we've showed this as well, that if you give diabetic models very high concentrations of folate, like 5 times what they should be getting, you can completely eliminate the neuropathy. And so this has been shown in several animals. We have human clinical trials going on right now. The trick is to blend it differently so you can lower that dose. There's one study that came out of Iran very recently that wasn't an RCT, but they just had diabetic patients. They put them on 800 micrograms of folic acid, and it increased their nerve conduction velocity. You can't tell what the way they scaled it, did the experiment, you can't tell how much of a rescue it was. But this is an example where diabetes has increased the requirement for folate than avoiding a comorbidity of diabetes. There are many more examples where we need to look into this. But it's also remarkable what the mechanism is. You don't need that much folate. Why does just the nerve become deficient? And why do you need so much folate to make that nerve function properly? You also have to think about the effect of pharmaceuticals can have on changing nutrient needs. And this is one of my favorite studies that came out of China. But enalapril, which is an ACE inhibitor, was used in an RCT to prevent stroke. And folic acid significantly affected the efficacy of that drug. Very high dose. Even if you didn't give folic acid in combination with the drug, that drug itself affects folate status and folate metabolism. And so we think as we age, we have a population on pharmaceuticals. What is that doing to nutrient requirements? And you're seeing a lot of pharmaceutical companies now blending their drugs with nutrients, mostly for the purpose of evergreening them, but also trying to avoid some of the comorbidities that you get associated with the drug itself. And again, aging is associated with more of these pharmaceutical uses. Another great example of tissue-specific deficiencies that can occur relate to cerebral folate deficiency. So your brain needs a lot of folate. There is a gradient between plasma and the CSF of folate, and you actively pump folate into the cerebral spinal fluid. And so you have this gradient of 1.5 of 3 to 1. And that gradient is critical to maintain because if this gradient goes away and you just have plasma levels, you see neurological symptoms of that. Things like depression and more severe mental outcomes. And so this barrier is critical to keep that folate gradient, which is ATP dependent, and a certain folate transporter there that allows active transport. But we also know that there's inborn errors of metabolism where you have mutations or mitochondrial depletion in that blood-brain barrier that don't allow you to keep the gradient. If you give them really high levels of folate, you can see clinical improvement in those neurological outcomes. Well, guess what? With aging, you also have mitochondrial depletion, you have ATP depletion, and as you age, that gradient dissipates. And it's been shown in trials that if you, again, give very high levels of folate to push it over that blood-brain barrier, you can see clinical improvement as well. So that's another example where during aging, you get these decline in networks, but you can reinforce with higher dose of folate. So when we think about half of the population having some sort of chronic disease, which you see now in most countries here, we have to think about very different issues. We have to think about stem cell biology, how to feed stem cells, how to restore function with nutrition, which is the big challenge. And one of the challenges here is time and money. And we covered this. My colleague Reagan Bailey and I held at the National Academy of Sciences about a year and a half ago an expert panel workshop where we brought in the very best people in AI and medicine, where they have built predictive models and knowledge graphs, where they can show with 80% certainty who's going to get Parkinson's and who's not 5 years before they actually show symptoms, just based on electronic medical records. We need this kind of technology in nutrition because these problems are really complex. And the problem is you have so many different foods, so many different nutrients and bioactives. You have so many outcomes to think about when you think about disease prevention. And you have to think of dose and time to onset and combinations, age and medicines. There's not enough time and money in the world to do these studies. There just isn't. We need innovations in AI, in these knowledge graphs, in these metabolic networks, predictive models that can then hone down to what are the important RCTs and how should they be designed. And we need, as was mentioned, biomarkers so that we can do high throughput combinatorial screening to really look at what is the right networks, what are the right combination of nutrients, what are the right targets that we move, get that data high throughput again to then inform clinical trials in the future. So that's what I have. Thank you very much.